Nice to be here with you, and I'm supposed to do something which is presenting our next speaker, which is Dr. Chris Ponting, okay? And his interest in miálgica encephalomyelitis grew from participation in the UK ME research collaboration, MERC, formerly known as the CFS ME Research Collaborative on CMRC. The DecodeME project evolved in 2018-2019 in a process during which PPI was embedded in every discussion and workshop, resulting in a full presentation production. He was the chief investigator of DecodeME and one of three individuals on its management group. The project was the world's largest ME CFS study with 27,000 UK-based participants, and from approximately 13,000 contributed to saliva DNA sample. And this was a very complex but very central study. Chris Ponting will be talking about, sorry, I didn't bring the, DecodeME genetics study. Okay, so I'm not sure. Hello, Dr. Ponting. Hello. Hello, Dr. Ponting. Can you hear us?
Hello from Scotland. And
we can see you and hear you. So the floor is yours. I just made your presentation. Thank you very much for joining us. The floor is yours. Thank you.
Thank you. And I hope that you can see my slides. Right, I'll take that as a yes.
Yes, we can.
Brilliant. That's excellent. So I'm delighted to be with you in virtually anyway today. Hope you enjoyed your lunch. Thank you so much for the privilege of speaking to you today, and thank you for allowing me to do so virtually. Today, I'm going to talk to you about the first results we have from the Genetics DecodeME project, funded in the UK by the Medical Research Council and the National Institute for Health and Care Research. It was a co-production, as we just heard, and that was critical, in my view, for the success of the project. So I'm going to talk to you about why we've looked at genetics in the first place, the process by which we got to the current state of results, the results themselves and what they mean, and what, in my view, should come next. So the main reason, in my view, why genetics is really important is to ensure that pharmaceutical companies in the future have sufficient evidence to hand for them to be confident to invest their funds on ME/CFS drugs and drug trials. They should be convinced by genetics. And I note here that 2/3 or so of FDA approvals have genetic support, and 92% of these have genetic support before approval. And so in this recent article, it was said that 40 targets, or 6% of FDA approved drugs, required genetics for the approval. So it's really important that we look forward to what I hope will happen and hope happens soon, which is that there are trials for interventions. The second thing why we do genetics is, and we look across the whole genome and all the genes, is that it's essentially unbiased and secondly comprehensive. And this is important because it may yet be determined that what's going wrong in people with ME is not rooted in known biology, but rather in unknown biology. And so if we stick only to known biology, then we would be overlooking the importance of various biological processes. And when a comprehensive approach is undertaken, then essentially the genetic can pinpoint what bodily system is implicated, whether it's the nervous system or the immune system or anything else. And that's important for ME, as we all know in the room that you're in. Everyone knows that there are very diverse symptoms of ME, and so this question of relevant bodily system is not yet confirmed. So how have we got to where we are? It's a project that has been going on for 5 years and more, and there were and are 3 objectives. First of all, we wanted to identify genetic differences that are statistically different in people with ME relative to population controls. And this would begin to explain biological mechanisms behind the disease. What we did not say at this point in time was that it would immediately lead to therapies and diagnostics. Secondly, we wanted to build a well-defined cohort of people with ME for future research, not really for us, but for everyone else. And so our cohort has been asked to consent, and the vast majority of people have consented to be recontacted for future research, and secondly, for their data to be shared at the individual level with other researchers around the world in academia and/or in industry. And that's the third objective, that we disseminate the data, and so that there will be outcomes from our study that were completely unanticipated. Now, the important thing that I've learned over this project is how important it was that we co-produced this project with people with ME. We understood their lived experience. They contributed greatly to the design of the questionnaire and how we discussed matters online with individuals, and led many aspects of the project. Our recruitment was based on people who are 16 years or above with a clinical diagnosis of ME by a healthcare professional, and people who lived in the UK only. And there's a very simple approach to what we've done, is to look at around about 500,000 places across our chromosomes and ask whether DNA variants, DNA letters, occur statistically significantly more frequently in people with ME over the population control. And if so, then what happens is that we draw a plot, which is here in the bottom right. This is not the real data, and the data points rise up above a horizontal line to generate what is called a Manhattan plot, and wherever these dots appear above the line, that is a genetic signal that is reported. So the key principles of co-production is that we share power, the research is jointly owned, and we work together. We include all perspectives, and we make sure that the research team includes all those who can make a contribution. We respect and value the knowledge of those who work together. Everyone is of equal importance. And there is reciprocity. There is not just a benefit to those who are researchers, but also those who are not. And that building of and maintaining of relationships is key to the sharing of power. And because there is a need to be joint understanding and consensus and clarity over roles and responsibilities, and important to value people and unlock their potential. So that's the key principles and values that we held to in this project. So I'm just going to, before showing some of the results, just show those who contributed to the delivery of the DecodeME project as listed in the preprint. And on the right was our original study protocol document. So, as I said, there were many people who signed up to the DecodeME project. 27,000 or so completed the questionnaire, 21 or 22,000 met eligibility criteria and were sent a saliva collection kit. And then an enormous proportion, 85% of people returned their kit, demonstrating the desire of people to contribute to research. DNA was extracted, their DNA letters read out and passed quality control for the vast majority of them. And the results that we've reported are those people of European ancestry. We would wish to analyze everyone's DNA, and indeed we're continuing to do so, but it's also important, very important, to match genetic ancestry between cases and controls. Otherwise, we would have come up with false discoveries. So that's why we did that. The cases that were defined are approximately equivalent in age, bottom left, and comorbidities, bottom right, with people who have an ME diagnosis, a G93.3 code in England. And so we believe that the cohort is fairly representative of the entire population of people with ME in the United Kingdom. The only exception is there is a substantial difference in the proportion of people reporting irritable bowel syndrome as a comorbidity. We also have responses from 15,000 people on a questionnaire 2, which includes their assessment as to whether particular treatments are positive or negative for their symptoms, and we'll be reporting on that as soon as we can. But suffice to say, unfortunately, there was not a single treatment that was very positively received by very many people. On the other hand, there were several treatments that were reported very negatively, especially graded exercise therapy. So here are the results that we pre-printed on August the 6th this year. There are 8 signals that we reported in a variety of the analyses. Here, 6 of them are reported from one of the GWASs, the genome-wide association studies, GWAS1. So again, what you're supposed to take away from this plot, this Manhattan plot, is that every dot represents a DNA letter, and we're testing to see whether they are frequently different in their frequencies between cases and controls. And if they are significantly different, then those dots rise above this horizontal line. So what we've done here is to annotate the genetic signals by genes that lie nearby to those signals, and you'll see that they occur on a variety of different chromosomes across the whole genome. We haven't yet reported on the X chromosome. So there were 8 regions that are statistically associated with ME status across the 4 studies that we presented there. And there will be evidence that I'll show you in a minute to say that they are relevant to the nervous system, the CNS, and the immune system. We found no evidence whatsoever that there was any sex bias in the genetic associations. We did test, but there was no statistical evidence for that. And 4 of the 8 signals did have statistical replication in data from national biobanks, but these associations were quite modest. So we were pleased to see that, but they weren't as strong as we would have hoped. So to do with the nervous system involvement, the study here was that we used an algorithm called MAGMA, which essentially looks at whether the activity of genes that occur in these genetic signals are more likely to occur in particular parts of the body. And I think there were about 55 or so different parts of the body that were tested, and 13 came out as significant, and all 13 of these were brain tissues. So what this does is to say that it is highly likely that there is nervous system involvement in the genetic risk factors of ME/CFS. So here's one example on chromosome 17, one of the 8 signals. This lies the signal, which is, as you see, the data points in triangles that lie above the horizontal line. This signal is just to one side of a gene called CA10. And what was really interesting for us here is that exactly the same signal was found in a different study, not a study of ME, but a study of chronic pain. So what has happened here is that the genetics of ME is speaking back to us as researchers of the chronic pain that people often report in ME. And so that closed the circle, really, and began to explain the genetics of some of the symptoms of ME. There is, we believe, an immune system involvement, and I'm showing on the right-hand side 2 signals that likely relate to the ability of people to respond well to infection. So on the top is a region of chromosome 6, which had a large signal between some histone genes, the H genes on the left, and the B genes, or tyrosine genes, on the right. And we believe that it may rely on the regulation of this gene here, BTN2A1, and our belief on that comes from a new observation that it is this gene that appears to have the most significant burden of rare variants that are deleterious among all genes in the genome, using UK Biobank AstraZeneca results. On the bottom right is another region of the genome on 13, where there is another signal. And this was very strong among people who had reported an infection just prior to onset, and is just next to a gene, OLFM4, whose function is in neutrophils in dampening down the innate immune system after infection. And I should have said the butyrophilins work as a sensor of bacterial infection. However, the DecodeME results do not immediately implicate single genes. What we have to do, and this is done in all such studies, is to use other data to implicate single genes, to prioritize them. And the way that we do this is to ask whether the ME genetic signal, which we're indicating here on the top line, is overlapping the genetic signal for a gene's expression, its activity. And if they do overlie one another, they superimpose, then they do something called co-localization. They are co-localized, and that provides additional evidence that it is the gene that contributes to ME genetic risk. So what we've done is to look at all of the genes in all of the 8 regions and use all of the expression data that's available from a resource called GTech and ask ourselves, indeed, whether the genetic signal for ME co-localizes with the genetic signal for gene activity. And where that co-localization happens, it is represented by larger circles here, the genes on the y-axis and the tissues on the x-axis. Green is where ME/CFS genetic risk is positively correlated with gene expression, whereas blue indicates where increased risk is associated with less gene activity, less gene expression. And we can also play this trick not only with genetic activity, but we can ask whether the genetic signals of one trait, here ME, co-localize or coincide with another, as we did previously with the CA10 chromosome 17 example. And so we were thereby able to come to the conclusion that there was no evidence for co-localization of ME genetic signals with any other human disease trait like major depressive disorder, except for the chronic pain example that I've already discussed. So this is the chronic pain example where essentially the DecodeME ME genetic signal in green co-localizes with the multi-site chronic pain signal. And we've plotted them on the same x-axis scale. So what are the implications of these initial results? The main implication is that genes do contribute one part of the puzzle. But what it doesn't say is that if people have these genetic differences, then they definitely have ME. It also doesn't say if they don't have genetic difference, that they won't get ME. And that's because DecodeME has only found a very small subset of all of the genetic risk factors that have yet to be found. And what we have tried to message from this project all the way through is that the project's results would not immediately lead to a treatment and testing. Rather, it would kickstart and accelerate future research, narrowing down what are the genes that, and their variation that contributes to ME and how, and also to focus our minds in the field to how we can accelerate towards those effective treatments and diagnostics. There is more to come. We are continuing our analyses. We haven't yet sent any manuscript to a journal. We're doing gene-specific work on using something called GATE. We've done some dominance GWAS, if people know what they are, and we've recently produced our final HLA results from associations and chromosome 6. We have done this analysis to compare with genes that carry a burden of rare variants, and we, in collaboration, are doing some more fine mapping. And we are carrying on our pledge to do more work on chromosome X, the mitochondrial genome, and potentially structural variants in the future. It was very heartening to see in work that was preprinted after ours that fibromyalgia as a risk was found to have genetic overlap with ME at two loci, the OLFM4 locus that I presented earlier, and the RABGAP1L locus. So essentially, some of what we have found replicates in a comorbid condition. And others, particularly people from Precision Life, have already identified overlap in the genes between long COVID and ME. So essentially what we have here is evidence for shared genetics among infection-associated and other chronic illnesses. They are not going to overlap completely, but there is indeed overlap in their genetics. As I said earlier, there is a data sharing process. It's all online. Happy also to talk to people individually. If you want the data, you apply for it. It goes to a data access committee, including independent members. Most applications are successful and data is shared. So what next in our view? Well, one of the things we would love to do is to take advantage of the 18,000 or so DNA samples that we have still in our freezers, and not just read out 500,000 places in our genomes, but all 3 billion places in our genomes. That's whole genome sequencing. That's an extraordinarily ambitious goal because it's very expensive. It would cost, for example, of the order of £20 million. Nevertheless, we do believe it has substantial benefits. And so we have teamed up with long-read technology leaders, Oxford Nanopore Technologies, again, the Charity Action for ME, and colleagues at EMBL EBI near Cambridge to plan this study in the hope that ultimately it will be funded. And that is because we believe that rare DNA variants will be able to shortcut the process to finding mechanisms of disease and effective treatments. And only rare DNA variants can be studied using sequencing and not the DecodeME approach previously. And ultimately, this same approach, this whole genome sequencing long read approach, can read out larger changes that are structural rather than point. And the same technology can read out DNA methylation. So we can answer these questions if we were to have whole genome sequencing using long reads of thousands and thousands of cases and compare them with available controls. So thank you so much for your time and the kind invitation to come and speak with you today. And I hope you're having a lovely time there in Porto.
Thank you, Dr. Ponting. If that's all right with you, we'll have a couple of questions from the audience. Please raise your hand. One first, and
thank
then
you, Dr. Ponting, for the really nice overview. I had two somewhat related but hopefully quick questions. The first one being, with the mutations that you got, or I guess with the differences that you're seeing in the genome, naturally that should lead to some sort of mutation functionally in a protein thereafter. So have you, or can you speculate on what those outcomes might be? Like, are these null mutations, hyperactivations?
So I'll answer that question first. So these are not known mutations. They're not known to affect any other human trait or disease apart from the chronic pain example I showed. They are not also of large effect. Rather, they are of very small effect. And it could be argued that their small effect means that they're not important. But that is not the case. And that is because even though they will have small effect, when you can intervene, ultimately, pharmaceutically, you can have major effect, a large effect. So even though these might pinpoint the regulation of the abundance of proteins, which is what we believe, in only a small way, if later on pharmaceutically you can knock down the whole protein or inhibit its function, that in theoretical terms would more likely be effective.
And if I might just follow up with that as well, there's been some interest in doing RNA sequencing or looking at those particular proteins that might be related to those DNA changes. Could you also maybe talk about how these certain mutations might then influence things like RNA sequencing?
Yeah, thank you. And one of the principles that we used for trying to determine which of the we prioritize is absolutely using RNA sequencing from the GTEC project across all of the different tissues. So we've already used that, the genetics of gene expression, to try and prioritize genes. But I'd like to say one further thing, and that is among all genes that are differentially abundant in mRNA or protein, some of those, probably most of those differences between cases, ME cases and controls, could be downstream of the cause of disease because of people's understandable inactivity or their drugs or other factors. And so when we find in our studies differences in molecular abundance, they need not immediately suggest to us that they represent the ultimate cause of ME pathogenesis.
Okay, we have already the two last questions. Yours first, and then I have an online one. Sorry, we are running late. That's why I'm being.
Hello, Professor Ponting. It's Rowan Gardner from Precision Life. Thank you very much for mentioning our work. Chris, you talked about the importance of co-creation of the data. Can you talk a little bit about how we continue to measure how well the participants in DecodeME are involved, inspired, enjoy participating in the research, and what your plans are in that area?
Thank you, Rowan. I hope you're enjoying your time there. We've just had a meeting with Precision Life just an hour ago, which I very much enjoyed. Your question is about co-creation and co-production. One way we measure the satisfaction of people contributing to DecodeME is the rate at which people have withdrawn from the study. And that is a very small proportion, and it happens in all studies. There have also been a relatively large number of people who have changed their consent to allow their data to be shared with others because they've understood, we believe, the value of this study ultimately. There is a paper being written, not by me, but by the PPI members of DecodeME to explain their views as to the benefit value that has emerged from the way in which we have co-produced this project.
Right, thank you. Dr. Ponting, one final question. Does the study eliminate the possibility that the nervous system genes are affected by the illness itself? Thank you.
Thank you. So no, we're not able to demonstrate a negative in that regard. What we have looked for is whether the genetics of ME and the genetics of all other diseases that we've looked at, including those of the central nervous system, can be explained in exactly the same way, genetically. And the answer thus far has been no. So no genetic overlap between ME and other diseases such as major depression. But what we can't say is that there is reverse causation or complicated interaction between genes and our environment, because ultimately that will have happened, and we think that because it has happened in many diseases.
Thank you, Dr. Ponting, and thank you once again for accepting to talk with us today.
It's a
Bye-bye.
privilege.
Thank you.
Thank you very much.